Southern California, and Senior Medical Director for Clinical Adoption at Cognoa. She was the 2018 President of the American Academy of Pediatrics, and her work includes integration of behavioral health and technology into primary care practice. Dr. Kraft received her BA at Virginia Tech and her MD and pediatric residency training at Virginia Commonwealth University with an MBA from the University of Cincinnati. It's my pleasure to introduce Dr. Kraft, and I'll let you take it away.
Thank you so much, John, and thank you, Pramila, for the opportunity to present here. I'm going to be talking about streamlining the current autism diagnostic pathway. Let me get my presentation up here and together. So we'll go ahead and get started. I'm representing Cognoa, which is a pediatric behavioral health company developing digital diagnostic and therapeutic solutions with the goals of enabling an equitable access to care, improving the lives of children, and really looking at those areas where access to care and quality of care is an issue, and to be able to use modern technology to improve that access and to improve that quality. Cognoa was founded in 2013, and their artificial intelligence-based technology originated with studies conducted out of both Harvard and Stanford University. Dr. Dennis Wall was the primary in all of that. I'll be talking about Candice DX, which is actually the first marketed product that Cognoa has out there. And it gained FDA marketing authorization as a digital aid to diagnose autism in young children, and really designed for primary care practitioners. So let's get started. And John mentioned this as well, too. That patient pathway from initial parent concern to the average age of diagnosis is way too long, and it's been too long for a long time. The CDC started its Act Early and Learn the Signs campaign in the early 2000s. And despite all of the awareness and all of the treatments out there and the explosion of access to resources to therapies and ABA, the average age of diagnosis has not moved the needle in 20 years. It's still at 4 years and 3 months. And we also know that the early intervention window is defined as that time frame when intervention has the greatest effect on long-term outcomes. And that's going to be between 18 months and 3.5 years. The earlier, the better for intervention. From a primary care physician perspective, this is what has always been and has always happened. And that is that child that you have seen from the time that they're a baby to an older age infant to a toddler comes in with a parent, and that parent will say, I am concerned about my child's development, or about their speech, or about their interactions. And what happens in the primary care space is the differential diagnosis of that concern is vast. And so what has happened over the years is that primary care has said, okay, we will refer you for that specialist evaluation. The problem is that there's a huge shortage of specialists. And so the wait to get in to see a specialist is anywhere on the average between 6 months and 18 months. And that shortage and the time-intensive evaluations are factors that result in those long wait times and delays in diagnosis. So the Candice DX algorithm integrated that training data and clinical guidance to look at the maximally predictive features of autism spectrum disorder. Using tools that exist, such as the ADOS and the ADIR, specialists in expertise with autism specialists were consulted during the development to look at what did they can look at as and figure what were the most important aspects and the most important questions of these particular tools. And what was it that they observed in the children that were most predictive of an autism diagnosis? So the algorithm was designed with this data in mind, and it was trained on thousands of sets of young children going through that autism diagnostic process and typical children, so that the algorithm could really juxtapose what that clinical scenario was and begin to start to separate out who had autism and who did not have autism. Candice DX is intended to aid in the autism spectrum disorder diagnosis in the primary care setting. The trial was done on children ages 18 months through 72 months at risk of developmental delay based on the concerns of a parent or a caregiver or a healthcare clinician. It is not intended to be used as a standalone diagnostic, but as an adjunct to the diagnostic process with those primary care clinicians who know that child and that family and what that child has been through throughout their life. There's no contraindications to using Candice DX. And this is the way that it is being used and proposed being used. Candice just launched in the beginning of November, and so we are beginning to see its use out in the field now. Your clinician prescribes Candice DX. It is a prescription diagnostic based on a parent concern or a clinician concern for developmental delay. There are 3 inputs that go into the algorithm, and these inputs come from parents or caregivers, they come from the child themselves, and they come from the clinician. So what happens is that once the Candice DX is approved by that child's and family's insurance company, the parents are given a link to download onto a smartphone or onto a tablet. And the first thing that they've got is a caregiver questionnaire, 18 to 21 items based on the age of the child via that mobile app. After they've answered those 18 to 21 questions, they are then prompted to upload 2 videos of their child. That first video is face-to-face, so that our video analysts who are master's degree level trained or above can look to see, does that child make eye contact? What is that child's speech like? What does that child do in terms of responding face-to-face with a caregiver? The second video is of that child in a play environment. And what's being looked at is that child doing typical play. Are they playing with a car, for example, and running it along a street? Or are they spinning the wheels and looking at it? Or are they lining up cars? So we're looking at typical versus atypical play. And in a play environment, you can look for stereotypic movements. You can look for toe walking. You can look for hand flapping. All of these things are then coded, and that becomes the second input. The third input comes from the clinician themselves, where they do a questionnaire, either in person or via telehealth, to ask specific questions based on that child's development that are somewhat focused towards autism spectrum disorder. So after the three inputs are collected, then the algorithm analyzes those inputs, and there are 3 outputs that come out. That child is positive for autism, negative for autism, or there's an indeterminate output. And that result to report is then sent to the primary care clinician, who can sit down with the family and go through some actionable items on what to do next. This is just a picture of what the caregiver mobile application and questionnaire looks like. So the questionnaires are easily answered. There's 17 to 21 of them based on the child's age. And after they answer the questionnaires, you can go on to the third screen there, when it shows the video instructions. And this is a schematic of where the output is for that primary care clinician. Remember, at that concern, traditionally, we have had this vast differential diagnosis with no way to try to figure out what's going on with that child. Instead, what Canvas does is it gives us a positive reading for some kids, a negative reading for some kids, and an indeterminate reading for some kids. Now, you will note the size of the bars on here, and it looks like indeterminate is the largest category, which in fact it is. 68% of those kids are indeterminate, but a third of the kids actually do get a positive or a negative output. And all 3 of these are actually actionable in a primary care space. I'll explain that in just a minute. But first, I'm going to talk about the Candice DX pivotal study. This is really important to understand where the data came from, where the outcomes came from, and how we know this device could actually work as a diagnostic aid for autism in the primary care space. So the Candice DX pivotal study was multi-site, prospective, blinded, method comparison cohort study. The primary endpoints were a positive predictive value greater than 65% and a negative predictive value of greater than 85%. And the comparison was with a specialist evaluation. How did Candice DX do when that child went through the process of Candice DX and then they were seen by a specialist? The secondary endpoints were sensitivity and specificity of the device in the overall study population. There were 425 completed children in the study. They were ages 18 to 72 months, 14 sites throughout the country. And then we had central review panelists as well, too, that oversaw both the Canvas clinical result and the specialist clinical result. This is just a schematic of the study flow for the device. Children were recruited when their parents expressed a concern or that healthcare clinician was concerned about their development and consented to go into the study. The first thing that happened was they underwent the Candice DX diagnosis. And once they had an output for Candice DX, they were then seen by a specialist. And these specialists were developmental behavioral pediatricians, they were child neurologists, they were child psychiatrists, but people who generally spent a lot of time diagnosing kids with developmental disabilities. If the Candice DX diagnosis agreed with the specialist outcome, and a central review panelist looked and agreed with both of these things, then that study was completed for that child. If there was disagreement, if there's a disagreement between the Canvas DX outcome and the specialist outcome, if there was disagreement between both outcomes and the central review panelist, that child actually went to another special evaluation. And once all of that data was collected, the best 2 out of 3 was the result that was reported. Some of the inclusion and exclusion criteria. We needed to have caregivers who could read and understand the informed consent and have functional English capacity. There needed to be caregiver or clinician concern for developmental delay. So this was not done on just typical children. Male or female, greater than 18 months, less than 72 months of age. Caregiver needed to have a smartphone app and internet capability to be able to download the study app. And the participants had to be willing to be videotaped during the diagnostic assessment by the specialist clinician. We specifically ruled out specific criteria, one being that that child was already diagnosed with autism. Secondly, that the child had pre-existing seizures or behavioral health diagnoses or were deaf or blind. We excluded kids who had actual physical developmental delay or criteria of dysmorphic features or an actual diagnosed genetic syndrome, because we really wanted Canvas to represent the children who were typical looking, phenotypically those children who one would be concerned about autism. When you looked at the demographics of the study completers versus the US Census, the children in the Canvas DX pivotal study were broadly representative of the US population in terms of race and ethnicity and socioeconomic background. So when you broke down race and ethnicity, the kids in the Canvas DX study really mirrored the population in the United States. This is a really important point because we know that the ADOS and the ADIR and many of the tools were actually research tools that were studied on primarily young white male boys. So how does that translate when you've got a child of color or a child from a different ethnicity or a racial background or a girl? So it was really good that in this study, we had that race and ethnicity background that reflected who was in the United States. Interestingly, the educational attainment breakdown showed that the families in the Canvas DX study had, on average, higher educational attainment than people in the United States. But conversely, their family income was less than average in the United States. And that represents the reality for many of our families who have kids with developmental disabilities. You often have to have one parent out of the workforce to take care of that child. The pivotal study results, the primary endpoints were achieved. So the positive predictive value was at 81%, greater than that 65%. The negative predictive value was 98%. And then the indeterminate output was 68%. So 1 in 3 children got a determinate output, a positive or a negative for autism. The secondary endpoints were also met. So the sensitivity and specificity were measured with a sensitivity of 98% and a specificity of 79%. How about those kids in that indeterminate platform? I was going to circle back to why indeterminate is actually very useful in the primary care space. What we found in the study is that of those 68% of kids, 91% of them actually had a neurodevelopmental diagnosis. As it turned out, 20% of those kids actually did have autism, but there were many other diagnoses, everything from global developmental delay to intellectual disability to selective mutism to anxiety disorder that became part of what we saw in through there. Why is this important? This is important because traditionally, what would happen in the primary care office is that that pediatrician would have 5 to 10 minutes with that child, and the parent would say, I'm concerned about my child's development. If the pediatrician or primary care provider or nurse practitioner saw something that was concerning to them, that child would be referred on to a developmental specialist. If nothing was seen in that 5 to 10 minutes, often what parents would get would be, well, let's wait a little while. I don't really see anything wrong. Let's go ahead. And as it turns out, these are the kids who wind up in that indeterminate pathway. So it really gives us a very clear, actionable next step, even though that determination was not positive or negative. Because what we are looking at here is as Candice DX becomes integrated into the primary care practice, it could enable more efficient and informed care planning. Candice DX would go into the workflow of a primary care practice, somewhat like ordering an X-ray. So you're concerned about something with a child, you order an X-ray, you get an X-ray report back, and then you sit down with the family and you use the information from your clinical suspicion as well as the X-ray to make a diagnosis. Very similar with Candice DX, that you have a concern, you prescribe Candice DX, you get an output, and that output along with your knowledge of the family and your knowledge of the child can allow you to sit down and go through next steps with them. So that child who's negative for autism spectrum disorder, autism is ruled out with these children. And what you would do with that family is talk about what your next steps are. If it was a child, for example, who might have an expressive speech delay, well, you get them into speech therapy. If it's a child that you think may have a primary behavioral health disability, you get them into some mental health resources. If it's a child where they have a sibling with autism and parents are just concerned and the child is typical, you watch and monitor closely. What if that child has autism by Candice DX? Then you streamline that pathway to services. At this point, a primary care clinician can write for speech therapy or occupational therapy. They can put in an authorization for ABA therapy. Some of what we're working on right now is that connection between the diagnosis and getting services, because it's not linear regardless of who makes that diagnosis. But you can start there. The other thing that primary care clinicians know is they know their local early intervention system and they know their local school systems. And many of them take care of kids who are in that early childhood special education program. So the advantage of having a child diagnosed in the primary care center is that these physicians and clinicians know the local resources and then can follow up with these families to make sure they were connected to these resources and that the kids get the care that they need. What about those indeterminate kids? These are the kids that you will have to refer on to a specialist because you don't have a specific diagnosis. In some ways, that's a safety feature. In some ways, that's actually good because not everybody with a developmental delay has autism. But it's really a good thing to be able to say, you know, we need to go on and have further testing for your child. But at the same time, if that parent is concerned about their child's speech, you could get them into speech therapy. If they're concerned about some sensory issues, maybe some occupational therapy. And think again that these are the kids that traditionally have been told, well, let's watch and wait and see. These kids are still going to be getting services earlier because of that indeterminate output. So Candice DX has the potential to help primary care clinicians effectively diagnose or rule out autism spectrum disorder when used in conjunction with what they know about that child and family, the clinical assessment, so that intervention can be initiated earlier than the current average age of diagnosis. And with that, I'm going to end my formal presentation and ask for questions.
Thanks, Colleen. One quick question. So since there's an indeterminate readout 68% of the time, how does billing occur? So are all families billed for using the system, or is it only if a positive or negative readout will the parent be charged?
So the idea is that Candice DX would be prescribed and would be covered or not covered as a diagnostic test by an insurance company. So, again, using the X-ray as an example.
Is it a diagnostic test or is it a screening tool?
As a diagnostic test. So a diagnostic aid. So, like an X-ray, you shoot an X-ray on a patient, you might get a negative result, but that tells you something. So even with the indeterminate output, it's important to know, because indeterminate is not negative. We know that 91% of those indeterminate kids have something. So it does give some actionable information at the primary care level.
Okay. All right. Well, thanks for your time. I don't think there's any other questions that have come through, but it was a wonderful presentation. So thank you.
Thank you. Appreciate the opportunity.
Take care. All right. Bye-bye. Neuroinflammatory and serotonin signaling.
Yeah, I will go ahead with.
Got it? Okay, cool.
Okay, so these last three presentations will be our last basic science talks of the day. Dr. Blakely is one of our scientific advisory board members, and he will be talking about bidirectional neuroinflammatory and serotonin signaling, basic and translational perspectives. Dr. Blakely is a native of Columbus, Georgia, a professor of biomedical sciences in the Charles Schmidt College of Medicine at Florida Atlantic University, and the founding executive director of the FAU Brain Institute. Prior to his arrival at FAU, Blakely held the Alan B. Bass Chair in Pharmacology and Psychiatry at the Vanderbilt University School of Medicine for 21 years, and when appointed was the youngest to hold an endowed chair in university history. At Vanderbilt, he directed the Center for Molecular Neuroscience, the Vanderbilt NIMH Silvio O. Conte Center for Neuroscience Research, and the Vanderbilt NIMH Postdoctoral Training Program in Functional Neurogenomics. In 1998, Dr. Blakely and Dr. Elaine Sanders Bush launched the Vanderbilt Brain Institute and generated a nationally prominent PhD program in neuroscience. He holds a dozen patents for his gene discoveries, novel research models, and potential therapeutic interventions, and continues to strive to translate his fundamental research to novel therapeutics. He has served on multiple advisory panels and supported medication development for multiple companies. Welcome, Dr. Blakely. You can take it away.
Thank you, Pramila. Very nice introduction. I'm going to share my talk. Hopefully you can see that now.
Yes.
Thanks so much for the introduction and for welcoming me into the Brain Foundation and supporting our research. So today, I'd like to continue a bit from a talk I gave last year that some of you may have seen when I talked about a mouse model that shows ASD-like phenotypes and how we could ameliorate those, thinking about immune signaling. And so today I'm going to extend this a bit and tell you where we've gone with this work and how this may point to bidirectional interactions between serotonin and the immune system. Let's see, can I advance this? Of course not.
I think you should click on the slide. This is some new update. If you click on the slide, it might.
Okay, great, thanks. Many people know about serotonin signaling, and I'm not going to spend a lot of time talking about the anatomy of it, except at one small place in the talk. But most people know about it because serotonin signaling can be potentiated. by the use of antidepressant molecules that target the serotonin transporter. These are drugs such as Prozac and Lexapro and so forth. And early in my work, this involved cloning the gene, encoding the serotonin transporter, and working out fundamental mechanisms as to how antidepressants interact with this protein and how it's regulated. But a good bit of our work moved over into the autism space when we found that by in situ hybridization, when we looked at the expression throughout development, we were surprised to see the serotonin transporter mRNA expressed very highly in a mid-gestation mouse embryo. So this is middle of embryonic development for a mouse. And it suggested to us that maybe if there were deficits in the function or expression of this protein, this might lead to some developmental abnormalities. And certainly we know that serotonin plays a role in many behaviors, and therefore maybe a neurobehavioral disorder as well. Now, thinking about serotonin at the time, what I knew about serotonin in autism was hyperserotonemia. This had been a biomarker that had been described 60 years ago as of this year by Shane and Friedman, indicating an increase in about 25% of individuals with an autism diagnosis, showing as shown on the right panel in orange, and just one of many examples, a significant group of subjects who have this elevated amount of serotonin in their blood. And this serotonin that's in the blood and that's accounting for hyperserotonemia is coming from platelets. And platelets store 99% of the serotonin in the blood. And this serotonin is made in the gut. It is picked up in the circulation by platelets through the protein that I was mentioning before, this serotonin transporter, this antidepressant sensitive serotonin transporter. And the platelets express this protein. It's accumulated inside the platelets and stabilized there in secretory granules, and they circulate around the bloodstream and use that serotonin in various ways. Serotonin levels in the bloodstream are highly heritable, more so than height. And we know that they're influenced by both genetic variation in the transporter, as well as SERT, interacting proteins, and other 5-HT homeostatic genes. So given that SERT was one of the proteins that could control blood serotonin levels, steady state levels, because of packaging it into these platelets, we wondered whether or not there might be some genetic changes in individuals with autism in SERT itself. And so with Jim Sutcliffe back in in 2005, we published a study looking for rare genetic variation in multiplex families with ASD. And we found 5 coding variants, which was, these coding variants are very rare. And in the number of subjects we looked at, it was quite striking to find this many. What was even more striking about these variants is that all of them, I'm just showing 4 here, all of them did not eliminate the function or reduce it. They actually increased the function of the transporter. They made it hyperfunctional. That was something very new to me. And it suggested that there might be a contribution being made, at least in the individuals with these variants, in traits of autism. And we showed that in the report that the most common of these is Gly56 alanine variant at the top that you see there, tracked with rigid compulsive traits and sensory aversion. So we were quite interested to see if this in vitro phenotype, which was replicated in the subject's own lymphoblasts, would carry through if we expressed it in mice in vivo. And before I get to the mice, just let me segue for a second and show you the model that we have for this increase in SERT activity. And I show here just induced by IL-56, the variant that we've studied the most there on the right. SERT star is an increased activity state of the transporter. And we know that you can move between the wild type and normal activity of SERT to the SERT star conformation via the activation of an enzyme called P38 alpha MAP kinase. This kinase is a well-known target of stress sensitive, of stress, cellular stress and systemic stress. And one thing that can activate that pathway is the inflammatory cytokine IL1 beta. And we showed that that cytokine would stimulate SERT activity in cells and in ex vivo in brain preparations. So one idea was that IL-56 was inducing a state permanently that IL-1 beta induced in SERT transiently. And so how might that play out then in terms of behavior or in terms of physiology? And that's why we made a mouse expressing that variant. So in that mouse, what we showed on the left was that these mice were exhibited hyperserotonemia. At the time, this was the first mouse model that had replicated this blood biomarker in ASD. And we were very excited just with that, and probably just should have stopped and then, published it. But we had to do a few more things to look at the physiology and behavior. And one important thing that you see on the right is that when you pulse serotonin into the brain and you measure how fast it clears from away from the pipette where you puffed it, which is the descending limb of this curve shown here, you see that the Gly56 alanine, the wild type expressing mice, clear serotonin at a nice rate, but the IL-56 mice clear serotonin much faster, indicating that in fact the transporters in vivo, because the density of the transporters was normal, is likely to be in this SERT star state, clearing serotonin away from its receptors faster and more efficiently than a normal system should. We also found that the G56A or IL-56 expressing mice, their serotonin neurons also did not fire at the same rate as the wild type SERT expressing neurons, indicating that there were two mechanisms by which diminished serotonin signaling could occur, one via diminished excitation induced release and the other by faster clearance by the transporter. Now at the behavioral level, I don't have time to go through the number of behaviors we studied, but I just wanted to show you one, which is in this simple test called the tube test, which is a test in some studies where you've established dominance hierarchies as a test of social dominance. In this case, these are not animals that have been in those kinds of hierarchies. And what it really queries is social avoidance. And what you see in the plot below is that when you plot the number of wins or losses by animals entering the tube and meeting in the middle, and a win means that they exit the tube nose first, essentially either pushing the other mouse out or the other mouse backs out. The IL-56 mice back out, and they show this pattern of social avoidance. Now, if you manipulate that regulatory pathway that I described, this P38 alpha MAP kinase pathway, what you can do is actually normalize many of the behaviors that we show in our work. And I'll just show you one piece of physiology, which on the left you've already seen. That's the elevated clearance rate for serotonin in the SERT IL-56 mouse. If you treat animals with this P38 MAP kinase inhibitor, it's called MW150, you see that the rates of clearance are essentially the same. So this is a 1-week treatment once a day with this P38 inhibitor, and they tolerate this drug very, very well. Additionally, the social behavior deficit that I described is normalized. So the purple bar is just wild types interacting with each other, showing 50-50 wins for whoever we put on one side versus the other. And the out of 56 animals, again, do not win in this test. But when you give them MW150, these animals show a normal win frequency. It looks actually like they're doing a little bit better here, but on average, that didn't occur. It was essentially normalized. So now I want to get back. I want to move away from the animals for just a second and just remind you that I was speaking about this P38 MAP kinase pathway that puts the transporter in its hyperactive state, and that you could activate that pathway with IL-1 beta and IL-1 receptors. And we showed that we could trigger peripheral inflammation and get this change to occur in the brain within 30 minutes. And it's mediated by IL-1 beta in the brain. So we were interested to find at about the time that we identified our variant and began studying it, that in fact the IL-1 receptor pathway and P38 MAP kinase pathway were shown to be elevated at the transcript level in microarray analysis of temporal cortex by Karoli Mernix and Tony Persico. So this was very exciting to us that, in fact, we had walked our way into an immune regulatory pathway that seemed to be upregulated, and inflammatory markers have been reported many times now in association with autism, either peripheral or centrally. And so we've been interested in this conversation going on, possibly between the immune system and serotonin signaling, less from a mechanistic perspective, and perhaps that there's something endogenous is happening here that might relate to autism. So one thing that we did was, as I said, we created a peripheral inflammatory insult using LPS and asked whether or not serotonin clearance in the brain is accelerated. And on the left panels, you can see that if we inject saline, the curves pre and post saline don't change. But if you inject LPS, which will give us an increase in IL-1 beta in the brain, you see this shift toward this steeper curve and faster clearance, and it's quantified on the right. This change in SERT-mediated clearance is paralleled by changes in serotonin uptake if you measure tissue ex vivo, and that's dependent on the IL-1 receptor. So we believe the pathway that I just showed you in terms of activating SERT is in the middle of this ability of the serotonin system to respond to LPS. And here are those synaptosome studies. I'll skip on through this. But again, identifying this IL-1 receptor and IL-1 beta system, regulating the transporter as an endogenous pathway by which serotonin neurons can communicate peripheral inflammatory status to changes in behavior. So now, are there IL-1 receptors on serotonin neurons? Well, yes, there are. Here's an immunolabeling approach where we're looking at in the leftmost panel serotonin neurons in an area called the dorsal raphe that are stained for serotonin, and in the middle panel you see a stain that's highlighting the IL-1 receptor. And you can see when we overlay them, there's quite nice correspondence, particularly in the dorsal areas of the dorsal raphe. This dorsal lateral area is very rich in IL-1 receptors and one of the richest areas in the CNS. So then one question became, what happens, what do these neurons actually do? What are their targets? And might that tell us something about behavior? And this has been an activity that Paula Kurtzel in the lab has been working on for the last several years. And we're beginning to make some headway. We use a technique called CFOS labeling. CFOS is a protein that's induced by neural activity And CFOS positive cells can be counted to indicate how many of cells in a particular brain region are activated. Now, one question that we've had is these neurons on the left, these neurons in the dorsal raphe nucleus that are serotonin positive and positive for IL1 receptors, where do they go? Where do these neurons go? And can we show that they change the activity of those neurons? Now, one way to do that, we can't be following all serotonin neurons. Only ones we want are the ones that are IL1 receptor positive. And to do that, we've used a genetic approach where we've eliminated the IL1 receptor, which you see on the far right. So this panel just demonstrates the red spots indicate that these neurons in a mouse, we've eliminated the IL1 receptor just on the serotonin neurons. So now what we can do is give LPS peripherally, look centrally, and ask which neurons are responding to LPS and changing the activity of targets that these neurons project to. Now, this is a lot of data here, and I'm not going to go through all of it, but there's really just two bars to pay attention to. We've queried males and females at different brain regions for CFOS positive cells. And what you can see, let's focus on the females in pink there. You see that when we give LPS in the periphery, this region of the brain called the lateral habenula, you see fewer of these positive cells, indicating that this nucleus has been inhibited. And when we go over to the the two bars to the right, these are those neurons where we've taken out the IL1 receptor. And you can see that this effect is normalized. So there is an inhibition of lateral habenula neurons in females that is totally dependent on IL1 receptors in serotonin neurons and no others. The lateral habenula of a male, on the other hand, we see stimulation of the activity in the lateral habenula, and that is normalized by IL1 receptors, a striking sexual dimorphism that is underlying behavioral responses to peripheral inflammation, now acting on the brain on a discrete pathway overseen by the lateral habenula. And why do we care about that? Well, the lateral habenula has been implicated in the pathophysiology of autism. I just want to point one recent paper out from Gurman et al. That used magnetic resonance imaging to show that there's a significant increase in the volume of the habenula in individuals with autism. The lateral habenula is pretty interesting because it mediates reward behavior, and particularly aversive behavior to either lack of reward, but also sensory stimuli that are aversive and social stimuli that are aversive. It also contributes to behavioral flexibility and inhibition, some cognitive states, sleep cycles, and pain responses. So a number of these are characteristic responses of a peripheral inflammatory insult, but they're also seen in individuals with autism as well. Now, I've tried to tell you about how serotonin and its modulation, either the neurons or the transporter by immune status, can change behavior. If we know something about it, we can tackle it and see if we can normalize the physiology and behavior that's disrupted. But one other aspect of this is that serotonin, this is not just a one-way street. Serotonin neurons also regulate the brain's immune system. And so here what I show is just a little illustration from sorry, that's not spelled right. It's Ho et al., 2013. Neurons release a number of molecules that regulate the status of glial cells, and in particular microglia. It turns out serotonin suppresses microglial activation, which is a pro-inflammatory state, and keeps microglia in a state that's supporting normal neural health and synaptic function, synaptic plasticity, through such as secreting TRAC beta to regulate neural precursors in the hippocampus, which we know is makes a contribution of for antidepressant-induced antidepressant activity. So if we're not releasing as much serotonin in the case of the IL-56 mice, if we have hyperclearance of serotonin as shown with the IL-56 mice, if any way in which we're diminishing our serotonergic output, this is going to lead to microglial activation because of the lack of inhibition. One way we could test that is to go back to our mice that have the IL-56 mutation and do a little sampling, which is something we've just begun to do now with Brain Foundation help. And what we see is just this 1 point mutation. In fact, it's a very, very subtle change in the structure of this transporter, but just that alone is enough to increase inflammatory cytokine expression. Here you see IL-1 beta and IL-6. You get an IL-10 response as well. And this is now SIRT IL-56 mice, the mutant mice that I showed you before. And this is in the midbrain. We see it in other brain regions as well. But another interesting aspect of this is that we see it in the periphery. And so here we're looking at the spleen, and you see also inflammatory cytokines elevated. You also see enlargement of the spleen in these mice as well that's genotype dependent. So what I think is happening in this animal is a case where serotonin neurons are clearly built to respond to inflammation. They respond to it early in development. They respond throughout life, changes behavior, and the changes in serotonin signaling capacity also change the immune system. That's been known for quite some time. But now with this point mutation in one protein, we're a little cleaner in our assessment that in fact, that one output of these serotonin neurons is sufficient to do what I showed you in the brain. The source of this effect, of this serotonin effect and SIRT effect in the periphery may well have to do with the enteric nervous system, which has serotonin neurons, or other aspects of peripheral serotonin that we haven't begun to investigate. And that's part of what we'll be doing in our new studies with Brain Foundation support. So let me just wrap up now and give you a summary. So I've told you that serotonin is expressed early in brain development, and that's led us to want to understand how alterations in serotonin homeostasis contribute to neurodevelopmental disorders. Along the path of that, we identified rare SIRT mutations, and these are functional. They induce changes in SIRT activity, increases in SIRT activity that are seen with the endogenous transporter when we make mice, and that they're similar to what you get if you treat preparations with IL-1 beta. We look at this in transgenic mice carrying that variant, and we see changes in physiology and reversible ASD-like behaviors. We know that high IL1 receptor expression in brain serotonin neurons leads to the change in activity of a few other brain nuclei I haven't talked about, one being the lateral habenula, and that SIRT hyperfunction on its own, without any other stimulation, induces a pro-inflammatory state in the brain and periphery. And it really suggests this bidirectional conversation going on between serotonin and the immune system as a normal aspect of physiology of the brain and periphery, but is disrupted when you diminish serotonin output. And we just showed you one way to do it by changing SIRT activity. So with that, I'll stop sharing and see if we have any questions. Oh my, there we go. Okay. So thanks a lot. And let's see, I am going to go over to the Q&A and see if I see any questions.
one question.
Does SSRI reverse the behaviors? We haven't done a lot with SSRIs in the model. They would be predicted to do so. And in some work that Jeremy Veenstra Vanderwill has done with these animals, some of these behaviors, yes, can be normalized with SSRIs. We haven't done this extensively. It would be good to do so. One of the problems, however, in using SSRIs is that no doubt many of these serotonergic pathways are not, they're not things from a clinical perspective. We don't really want to block them all. We'd like to get to the ones that are in a hyperactive state. Now, remember, these mutations I showed you are rare. So thinking more broadly, it'd be better if we could go after ones that are hyperactivated. And I suspect that these would be because of immune activation, pro-inflammatory activation.
Can I ask a question?
You certainly can.
Yeah, I'm a clinical immunologist, so I was wondering what's the serotonin uptake stat of the congenital deficiency of the some of the genes, which is usually causing, like autoinflammatory syndrome, causing the over-production of the IL1 beta, or the deficiency of the IL1 receptor antagonist. Those are very typical autoinflammatory syndrome. What happens in these patients in your serotonin pathway?
Yeah, I think that's a really interesting idea. You know, the most that one typically sees in the literature is related to mood disorders and depression and high frequency of mood disorders in subjects who have chronic inflammatory signaling. And you know, some of these occur during during adulthood. When this occurs early in development, I think that there'd be other changes associated with behavior that I would predict would have a serotonergic component to them.
Mm-hmm. Yeah, because they're very difficult patient, and like omid type patient, and very sick, very irritable, like a serious meningitis type symptoms. So I was wondering, but I'm not aware of any of the report for the serotonin mechanism in these patients.
Yeah, and when they are reported, like I said, they most often have been evaluated in the context of mood disorders, certainly irritability, anxiety, those kinds of things. But you know, the kinds of social behavior changes that we see in our animals, we also see repetitive behavior in them as well. And we see poor communication between pups and mother as another hallmark of their behavioral changes that is seen in a lot of animal models of ASD.
So the treatment we use anakinra, that is like a human recombinant IL1 receptor antagonist. Does it work? That is human, but I think the mouse IL1 receptor antagonist, I think it's available. Yeah, I wasn't sure. I'm curious if it's available.
We haven't tried that, but we have genetically eliminated the IL1 receptor and done that just in serotonin neurons. And it definitely prevents the ability of a peripheral immune insult to affect CNS serotonergic signaling. But in terms of the IL-56 case, we haven't looked yet.
Okay. Yeah.
So let's see, I have a question about how prevalent are the certain mutations in a general population? Is there evidence for increased generalized in humans with certain mutations? So the IL-56 variant that we worked on is the most common one, and it's at about 1% or less. And so, you know, there just has not been an attempt to try to gather enough of a population to do, you know, those kinds of experiments. It would be great. The other mutations I talked about are, private ones, essentially. They're in individual families. And then the follow-up, what is the mediator between serotonin and inflammation? How does it talk to the immune system? Why use serotonin for modulation? So there are serotonin receptors on microglial cells, and our current hypothesis is, in fact, that neurons talk to microglia through many different signaling molecules I showed you, and serotonin being one of them. There's a lot of interest in how SSRIs may reduce inflammation in relation to neurodegenerative diseases. That's caught the attention more, I think, than people looking into autism or in depression. But I think it's quite an interesting dimension, and we're trying to push this for funding from the NIH. Why use serotonin for modulation? I'll wave my hand and think teleologically here, but I think that, the kinds of things that need to happen, and this has been said by psychoneuroimmunologists for a long, long time, is that you need certain behavioral states are protective in the context of inflammation or infection. And the things that you need to change by way of your behavior, your metabolism, and your physiology are all under the control of serotonin signaling. And the serotonin system is very broad, and it can control many different behaviors in parallel. And so one important outpost are these neurons to affect a number of different behaviors, like stop eating, get away from people so that you don't infect the rest of everybody else around you. Get some sleep, conserve your metabolism. All of these things have serotonergic modulation available and use. And I think the immune system, we have to think about the peripheral immune system as going after discrete circuits to change behavior, not just to go in and make people sick. So I think we're at time. I guess I can move on to our next speaker in the session, who needs no introduction, so I won't give him any. No, he's my friend, Dr. Aurelio Galli. He's director of the gastrointestinal biology research in the Department of Surgery, and also holds an appointment in the Department of Neuroscience. Previously, I got to know Aurelio because together we were on the faculty at Vanderbilt. He was in molecular physiology and I was in pharmacology. Aurelio has dopamine transporters, but he studied other things besides that. And he's an expert in cellular physiology and molecular structure of these proteins. And then in recent years, looking at their contributions to behavior, including autism. And today he's going to tell us about flies, I think, and how you take mutations that are related to autism and learn some interesting things by studying them in an invertebrate. And probably also you'll talk a little bit about mice too, I suspect. Welcome.
Thank you so much, but I cannot share the screen.
Pramila, can you make it for him to share?
You can try again now. Be able to share.
Let me see.
Looks good.
Do you see the PowerPoint presentation, or let me see.
Yes.
Yes.
Do you see the comments, or you see just the PowerPoint presentation?
Just the presentation.
Okay. All right. So, well, thank you so much to the Brain Foundation and Randy for the kind invitation. And our group has a really strong interest in understanding how disruption of neurocircuitry or molecular mechanism can lead to neuropsychiatric disorder. In particular, we are interested in understanding the role of dopamine dysfunction as a complication or participating as a mechanism in the etiology of amphetamine abuse and for the purpose of this talk in autism spectrum disorder. So for example, we are interested in understanding how amphetamine hijack particularly neurocircuitry in the brain in order to cause dopamine dysfunction and amphetamine abuse. We're also interested in understanding how single nucleotide polymorphism and de novo mutation in the dopamine transporter gene can disrupt dopamine transporter function, causing impairment in dopamine neurotransmission that we phenotype with specific behavior. And we use an array of tools to study these genetic variants, from structural biology to high-speed camera to look at the behavior of these flies. So dopamine dysregulation has been implicated in a number of neuropsychiatric disorders, ADHD, schizophrenia, neurological disorders such as Parkinson's disease, and lately also to autism spectrum disorder. In fact, some of the mutants that we analyzed in the past in collaboration with Randy Blakely has been found both in patients with attention deficit hyperactivity disorder and also in patients with ASD. So why ASD and dopamine dysfunction? There are a lot of traits that relate to autism spectrum disorder that points to dopamine dysfunction, from repetitive behavior, lack of rewards for social interaction, hyperactivity, anxiety, and so on. So in the scientific community in the last few years, starting to publish paper looking at the dopamine transporter dysfunction and his association with autism spectrum disorder. There are not many papers that have done that, but like I said, in the last few years, particularly our group in collaboration with Randy too, has published a few papers regarding this subject. So just for giggles, there are two main dopaminergic projections. One is from the nigra to the striatum, and the other is from the ventrotegmental area to the nucleus accumbens and prefrontal cortex. What is important for this presentation is that the dopamine transporter is the main protein clearing the extracellular dopamine level once dopamine has been released. And really, it's been a renaissance for the dopamine transporter lately, because we discover a lot of very interesting mutations related to neurological disorders such as Parkinson's disease, of infantile early dystonia, and neuropsychiatric disorders such as bipolar disorder, ADHD, and ASD, that give us really the opportunity to apply all the knowledge that we have acquired in the last 10 years about the dopamine transporter to understand really how this mutation disrupt transporter function and leads to specific behavior. So this is the topology of the dopamine transporter, is 12 transmembrane domain, and up to now 5 mutants has been characterized in the dopamine transporter that has been associated with autism. They're all in critical structural domain of the transport. One is the N-arginine for tryptophan at position 51 in the N-terminus of the transporter. That is an important structural domain regulating transporter function. There is a deletion, an in-frame deletion of an asparagine at position 336 on the third intracellular loop. This is another mutation that is very, very important, because the third intracellular loop is one of the loops regulating the intracellular gate of the transporter. The transporter have 2 gates, one extracellular, one intracellular, that are controlling and regulating the movement of dopamine across the plasma membrane. There is a alanine to a valine on the top of transmembrane 12. This was a mutation that was first discovered in a bipolar patient, later on at Vanderbilt and described by Randy and me for attention deficit hyperactivity disorder. And again, with the Vanderbilt patient in autism, again, characterized by Randy and myself. And finally, the first de novo mutation on the top of transmembrane 7, it is a threonine to a methionine. So when we look at the dopamine transporter, the physiological function of this protein is, how I said, is to clear extracellular dopamine for the extracellular space and move it inside. And typically, all this variation in pair transporter function, at least the one that we analyzed, by 2 different modalities. The first modality is simply that the transporter goes on the plasma membrane, but is not capable to reuptake dopamine anymore, right? And so there is an impaired dopamine uptake. And the second modality that is much more interesting for me, because I'm a biophysicist by training, is that now the transporter run backwards, right? For reason that can be signaling pathway or structurally, now the transporter binds dopamine from the inside, the neuron, it pumps dopamine outside, increasing it therefore extracellular dopamine level, and is what we call anomalous dopamine efflux. And this has been noticed for the first time in the A559V mutation, and later on in the T356M mutation associated with autism. So the proband is a male, has been classified as with autism spectrum disorder at age 5. And one of the important things about this threonine at position 356, it is highly conserved across phyla, from human to C. elegans. And the reason why it's so conserved, because he lies within the permeation pathway of dopamine and the co-transported ions, in this case, sodium. So you can imagine that any spatial constraint that the mutation threonine to methionine can produce can impair, of course, the function of the transporter. And so like I said, we use a lot of structural tools from molecular dynamic simulation to structural biology, and also electron paramagnetic resonance spectroscopy to measure really how these mutations are impairing dopamine transporter function by looking on a structural perspective. So in collaboration with the Samu Chara by Vanderbilt University, we do EPR, so electron paramagnetic resonance spectroscopy. So we engineer cysteines that are spin labeled, and EPR allow you to monitor the distance of these cysteines that we use it to look how the intracellular gate or the extracellular gate of the homologue, the bacterial homologue of the dopamine transporter is. And we look how these mutations are affecting the movement of the intracellular gate or the extracellular gate of this protein. And so without going into much details, what we saw that the intracellular gate was not really affected by the mutation, but we were looking at the spin label that we position on the extracellular gate of the transporter in the presence of sodium, the co-transported ions, and the substrate, in this case leucine, right? In the wild type, there was a rearrangement of the extracellular gate that was going in an occluded pore, right? So the transporter was trying now to move dopamine from the outside to the inside by closing the gate. But when we look at what was happening on the mutant, we saw a higher probability of these two cysteines, the two cysteine label, to be at higher distances, meaning that now the gate was opening to the outside. So when it was binding the substrate, moving the substrate outside the cell. So this was really the first structural fingerprint of what anomalous dopamine efflux was, meaning that the transporter, when it binds the substrate, spews the substrate outside. So we had to choose a model now, an animal model, to start to screen all these mutations. And of course, mice cost a lot. And these we were running on fume on these projects, right? So we choose Drosophila to express the human dopamine transporter or the mutated dopamine transporter in the right neurocircuitry, meaning the dopaminergic system. So we use the US GAL4 binary system now to express, like I said, the human DET or the mutated human DET in dopaminergic neurons of flies that are lacking the Drosophila dopamine transport. And so the first thing that we've done, right, is to look at the fly brain that you see over here. There are a lot of dopaminergic projections, you see them in red, and to start to figure it out how to study anomalous dopamine efflux. Again, the transporter running backwards. Before the work was done only in cells, and now we wanted to move, of course, to an animal model. So what we use is amperometry, is a technique that takes advantage of a carbon fiber that we position in any dopaminergic node of the fly brain. And this carbon fiber measures the level of dopamine by oxidation, right? It is represented as a positive current. So you can expect that if you have an anomalous dopamine efflux, the baseline recorded by this electrode will be higher than in the wild type. You will also expect that if you block the transporter, either with methylphenidate or other blocker like cocaine, this baseline goes down because you're blocking the constitutive dopamine efflux of dopamine. And this is what we notice over here, right? So you can see that you're already cocaine and the baseline goes down because you're blocking the constitutive dopamine efflux of dopamine. And we've done several studies characterizing structurally and the signaling pathway regulating the anomalous dopamine efflux, including we saw for the FFNV mutation, there was phosphorylation of the N-terminus of the dopamine transporter. So what does it mean, this behavior? The first thing that we did is to convince ourselves that flies was a good model to study neuropsychiatric disorder. And we asked the help of a very famous scientist, Charles Darwin, and in one of his books, The Expression of the Emotion in Men and Animals, said that even insects express anger, terror, jealousy, and love by their stridulation. And so we thought that we were on the right track. And so the first thing that we looked, it was how the anomalous dopamine efflux was influencing locomotion, because a lot of patients with ASD are also hyperactive, right? And so I want to show you a movie. This is video tracking of flies expressing either the human death or the human death that now has been mutated. And you can see that the flies with have the human death causing anomalous dopamine efflux, they are moving much faster. They're crossing the center of the cage much more, and they're grooming much more than the flies. They're just expressing the human death, and this is the path that the flies that are expressing the human death with the mutation are performing during this time. You can also do beam crossing to look at the circadian locomotor activity. So these are the wild types, so the human death over at the turn on of the lights and the turn off of the lights over 26 hours. And you can see clearly that the human death expressed in the 356M is completely hyperactive, almost similarly to flies that are not expressed in the transporter at all, and therefore have really high level of extracellular dopamine level. And you can quantify this over 24 hours to see clearly that the mutation causes hyperactivity in these flies. So another characteristic in autism that a lot of young people with autism spectrum disorder have a really high level of anxiety, with fear to be one of the most common subtypes of anxiety in autism. And so the question is really how to measure fears in flies, right? Animals, I'm a really outdoor person. An animal, I know that they fear under predatory pressure. And they fear to camouflage, but they fear also because they are scared, right? And the question was how to scare flies, right? And one time I was in my cabin where I pick a lot of mushrooms, right? And I saw one of these guys is a predatory wasp close to a nest of flies. They emit a sound like this. I don't know if you can hear it. And so the flies are scared. I'm sorry to turn off the sound because it was making me deaf. And the flies are scared simply because the predatory wasp is eating their larva. And so what we did, we recreate the sound of the predatory wasp in the lab. We use a high-speed camera. This camera can record 50,000 frames per second. This is a video that we recorded at 3,000 frames per second. And if you don't hear the sound of the predatory wasp, there is a red highlight of the cage that will tell you that the sound of the predatory wasp is coming on. And we're measuring freezing behavior in these flies, right? So you can see the flies move around. The video is being slowed down. The sound come on, they freeze, and they escape, right? And this is a natural behavior. Usually animal freeze, and then they look in the environment, they escape. And so this is the mutant instead. And you can see that the freezing time of this of the mutant is much longer than the wild type. You can even notice it by eye. So the mutant moves around. It really doesn't move anymore. Right? So you can quantify this average velocity of the fly every millisecond, right? And this is the wild type in black. The animal moves around, the sound of the predatory wasp comes on, the animal freezes, and then is escaping. Instead, if you do the same type of experiment with the mutant, the animal moves around, the sound of the predatory wasp comes on, the animal freezes, and it keeps staying in a freezing state. Finally, we wanted to look at social interaction in autism that are impaired, looking again at what anomalous dopamine efflux does to this social interaction. So we have to find ways to measure social interaction again in flies, and there are several ways. I thought that this would be a cool way to look. You know, if you look at the school of fish or flocks of flies or birds, They maintain social distances. They maintain social distances because they try to survive in the environment, particularly when a predator is around. This is a school of fish that's being predated by a duck in the back. You can see it over here. And one thing that you notice is that the school of fish compress and expand depending on the distance of the duck, right? And so this is a behavior that is being modeled to say that when the animal, the predator approach the flock, the school of fish, the school of fish will compress and then will immediately expand because they try to escape altogether away from the predator. So we thought that we could do the same using the flies, meaning that we can measure the social distances of the fly, the cumulative social distances over time before and after the sound of the predatory wasp. And so this is what we saw. So this is the wild type, so expressing the human death. And it's at the on of the sound of the predatory wasp, they compress and then they expand. And if you look instead at the mutant, first of all, they start from a compressed state, right? But at the on of the sound of the predatory wasp, they expand and then they compress. So they do the opposite of what the wild type are doing. And so what we are interested in is not only about the human dopamine transporter, but also other proteins, right, that participate in the signaling pathway that are regulating dopamine transporter function. As we talk, we are working on syntaxin 1A. This is a protein that regulates the ability of the transporter to reuptake dopamine. Today, I show data that parallel a mutation, the first de novo mutation in the dopamine transporter, that cause anomalous dopamine efflux. We characterize this mutation structurally, so we fingerprint what anomalous dopamine efflux occurs for this mutation, that is the opening of the extracellular gate, allowing the dopamine to be released outside. We also measure dopamine efflux, anomalous dopamine efflux straight in the brain of flies, and link this to impairment in specific behavior of the fly, including increased locomotion, increase fear response, impair social distances. Of course, the question remains, what is happening to more complex behavior that cannot be studied in flies? And so we have done a knock-in mouse, right? And we show in the knock-in mouse that we have impaired dopamine clearance, decreased tissue dopamine content, increased locomotion, impaired social behavior, and loss of social dominance. And you can see all the data published in a recent article in Journal of Clinical Investigation. I do have to say thank you to a lot of people, particularly Gabriela Di Carlo, Peter Hamilton, Aparna Shekhar, Jenny Aguilar, and Christine Saunders, Andrea Belovich, and outside the lab to Greta Galli, my daughter, that did some of this experiment, Kevin Errigan and Einar Mattis. Einar Mattis is an expert in genetics of flies, Mark Wallace, who together, the geneticist James Sutcliffe, and finally for the EPR to Assam Mucharab and Shruti Sharma, and for you all for listening to me. Thank you so much again.
Thanks, Aurelio. It's just amazing to see these conservation of these behaviors. Obviously, you know, a fly is not going to be a mouse, is not going to be a human, but we can trace these different systems, as you showed, you know, nicely with the fish and the flies, and the way in which we have protective behaviors ourselves together. It's just amazing. I have a couple of questions just of my own to remind me. And you know, I'm sorry, I should remember all of this, but in the fly model that you introduced us to, was that the human mutation in the fly background, or was it in the human background?
It was, so we express the human dopamine transporter directly in flies in dopaminergic neuron. So these dopaminergic neurons are lacking the Drosophila dopamine transporter. So quote unquote, the flies get humanized with the human dopamine transporter.
I see. And do you put the wild type in parallel as your control?
Yes. So
okay.
Yes, and we backcross this fly quite a bit. So.
And does the animal with the human dopamine transporter look, and just the wild type, does he look just, does he behave just like a wild type fly with a fly transporter?
You know, we have done this for locomotion, and they do, right? You know, the high-speed camera experiment are really complicated. So we don't have a parallel with the Drosophila dopamine transporter, but we always compare it to the one expressed in the human dopamine transporter. In terms of uptake and efflux, they are the same. So the one expressed in the human dopamine transporter are really similar in terms of clearance and response to psychostimulants, for example, to amphetamine, to what we have seen with the flies expressed in the human dopamine transporter.
So maybe with time, because these other domains that are probably divergent have regulatory elements in them, and you might find some really interesting things where the transporter is regulated, maybe not appropriately because of the sequence divergence.
Yeah, we are working heavily now on the microbiome to look how it's influencing transporter activity. Right. So.
Right. Yes, Dr. Galli has quite a nice program going on microbiome, and with his dopamine background as well. If anybody's interested in asking about that either now or later, I'm sure he'd be interested in hearing your questions. I have one more question about flies and how you use them. So a lot of people will turn to Drosophila because they can screen for mutants or screen efficiently, genetic stocks that have mutations in a whole bunch of things. Have you explored yet any of these genetic variants that might, if you put them together I think that your T356 mutation, this de novo mutation from individuals with autism, is a very powerful reminder. It's powerful evidence that the dopamine system can contribute to autism, whereas the rest are probably more risk factors and need other things to come together. So that was what sort of made me think a bit about some of these other mutations that might be interesting to put together. Have you put any together with yours?
No, but the T356M is actually found now in another patient in Cincinnati, and we're in contact with the doctor. They just swap the saliva and send it to us to look at the microbiome, and also in a new patient in Europe. So it's becoming a very interesting mutation to follow up in all these kind of things. In the mouse, for example, the one that I show in the knocking mouse has a difference in the fusobacteria in the gut, right? It has been linked to dopamine dysfunction and in autism. And we measure the microbiome in this mouse, and now we're trying to compare this with the patient. Dr.
Blakely, there is a question in the Q&A.
Okay, let's see. Can I find it here? Okay, what do we know about neuromotor issues in the mutants? How do we go from DAT to behavior response? Aurelio, you showed us hyperactivity. Mm-hmm. You also showed us repetitive grooming behavior. Mm-hmm. Any other motor responses that might interest this audience in relation to?
So in the knocking mouse, they are performing much better on the rotorod. That is another motor response that I remember now on the top of my mind. And the mouse is hyperactive too. I try to remember other things. In the social dominance test, they lose, like you show for your mutant and the serotonin transporter. Mm-hmm. That is about it that I remember that we've done. And the mouse is available to everybody. So whoever is interested in using this mouse, just email me, and I'm more than happy to send it to you.
So maybe, Ashok, if you have a more refined question about neuromotor issues, were you thinking about something in particular? But Ashok has another question about that microbiome. He wonders about specific metabolites that might be secreted that could be responsible, I think, for some of the behavioral changes that you're seeing.
What other metabolites do you have in mind?
Are there bacterial produced metabolites that might be affecting brain function?
Okay, so we didn't look at that. So the fusobacteria is producing butyrate, And we have a model in actually amphetamine abuse, because a lot of methamphetamine abuser has increased fusobacteria in the mouth. That is why they have gingivitis. And so when I was reading and finishing up this paper, hopefully soon, on butyrate and amphetamine and methamphetamine abuser, I discovered that in autism, it looks like this kid has low level of fusobacteria. So we got a knocking mouse, and this was the first thing that we've done, is to look at the fusobacteria in this knocking mouse, and they are low. We didn't measure butyrate because it's actually quite difficult to measure.
Well, that's going to be interesting to follow up. One of the things on the serotonin side, serotonin obviously in the gut affects the microbiome as well.
Yeah.
And we've been trying to get our mutation in a cell-specific knock-in, which is a little harder to do than the transgene or just a standard knock-in to do it conditionally. But I have wondered how much of the behavioral phenotype that we have is due to what's happening in the gut being transmitted to the brain, just like the immune function, right?
Yeah, in flies, butyrate influence a lot of this behavior. It's influencing courting behavior, is influencing locomotion. So we feed the butyrate directly to the fly, and we modify the behavior of the fly. Now, we didn't do it in the mouse.
It's an interesting story about amphetamine, and I understand why you would do that, given dad is the site of mutation. Let's see. I think that's the end of the questions that I see. I'll thank you again, Aurelio, for
thank you so much.
A very interesting talk and providing this insight, this evolutionary insight into how we study autism mutations. And now move to our next speaker. So, of pediatrics at Rutgers, New Jersey Medical School, associated with St. Peter's University Hospital. And her work has looked at neuroimmune interactions and epigenetic regulation in children with autism. She also has interest in immune abnormalities and peripheral disorders such as asthma and GI dysfunction. So it's a pleasure to have you join our panel, and I'll turn it over to you now.
Did you see the slide?
No, please open the slide and then hit share screen and then select.
Okay, share screen. Okay.
Yeah, and select the presentation.
Yeah. Oh, I hit the share screen.
Does it show you the presentation in the panel?
Yeah.
Just select that.
Let's see. You don't see that?
Not yet.
Okay. I think that I hit the. So that I hit the screen. Did you see?
Yeah.
Now we do.
Okay.
Now we're getting there. Yeah. And Harumi, you might want to turn your camera down just a little bit because we can only see you from the nose up. Perfect.
Okay. Yeah.
Perfect.
Sorry. Yeah.
And if you can please go to the slideshow. Oh, yeah.
Okay.
Thank you.
Yeah. Now it's okay.
It's good.
Yes.
Okay. Yeah. And thank you for inviting me, and also very generously funding this project. So this is a progress report of the project that funded by the Brain Foundation, and essentially focused for the innate immune memory biomarkers as a part of the neuroinflammation. So, So the Brain Foundation funding this project has two aims. Number one, I have been working on the monocyte cytokine profiles as a marker of the innate immune response. And we want associate with clinical characteristic of those cytokine profiles. And the reason why we looked for the monocyte cytokine profiles, that's as a marker of the innate immunity. Obviously, innate immunity has been thought about for some implications of neuroinflammation in other type of the neuropsychiatric conditions. And it has been worked as for the in other common neuropsychiatric conditions such as depression, schizophrenia, something like that. Then why the innate immunity and the innate immune memory was really implicated? All these neuropsych conditions, all the studies tried to implicate with one specific pathogens associated with condition has been all failed. So all the conclusion is really non-specific immune response, which is typically the innate immune responses associated with onset and progress of the neuropsychiatric symptoms. But then its question rises, why is the innate immunity, which doesn't have any memory, but a lot of condition once activated, then it's very lasting effect. So obviously the innate immunity doesn't have any memory for specific pathogen, just like if we have a measles, we have a very prolonged memory for measles. But then, about like 10 years ago, from that Dutch group, there is a concept of the innate immune memory, and which is essentially the epigenetic changes of the monocyte or macrophage populations. And those monocyte or macrophage and also other innate immune cells are so true. They're all later found out. There is an innate immune memory generated in response to fairly strong immune stimuli. And those changes were initially metabolic changes. And it's usually ATP, ATP production to more getting into the glyconeogenesis, then accumulations accumulate, which is changing to a histone modification, affecting the histone acetylase and histone methyltransferase changes. So essentially a lot of things associated with a mitochondrial functional change. Then also it's a prolonged case. It has been shown there is a change of the histone modification. I'm sorry, it says I misspelled. Histone modification markers is changed. And this is for the ASD. And so that's a part of the reason we check first the monocyte cytokine profile, and then how associated with clinical feature. Then how these condition was also any associations, monocyte histone modification markers. And then in the selected patient, we're going to do the super enhancer chip sequencing. Then which target to be used for super enhancer chip sequencing, we will decide based on the expression of the histone modification markers. Then M2 is for the more likely the quick assessment of the serum markers, which can be used for screening of those changes. Then previously you have done this deep sequencing of the microRNA in serum. And the reasoning why we did that, because the major source of the microRNA in the serum is a platelet and monocyte macrophage. And also the microRNA as secreted as a form of the, it's like a cyst with like a, it's a fairly stable and secreted as like increased in the endosome. So that's the reason why the microRNA, because we did a long time ago, we did the serum cytokine levels, and those are not very helpful. So we wanted for the little more reliable, like a serum marker. So these are two purposes of the our proposed study that the Brain Foundation funded. And then we tried to do the this. We haven't completely finished, but so that it's some of the progress report. Some of AM2 is almost finished. So okay. So the initial study for the associations between clinical features and monocyte cytokine profiles. So that was we did that because the previous studies and also the more newly recruited. So we have done that about like close to 150 in ASD subject. And some of the clinical association, it was significant is GI symptoms and TNF-alpha production. Then also for the IL-6 and IL-1 beta, that was more associated or fluctuating behavioral symptom. It's categorized as PANS, but I don't think the ASD patient has a real PANS. It's more likely the post-infectious neuroinflammation, and it does show the symptoms like anxiety, OCD, anxiety, irritability, and worsening hyperactivity type. And seizure disorders, which is not surprising, at least our data showing some association of the IL-1 beta production by monocyte. And we just made sure those associations are not associated with the medications they're currently on, because recruited patient is not really ruled out with the use of the medication. And also it wasn't affected by age, gender, and the other covariables we checked. And also we didn't see the significant association with the autism severity by ADOS. So then for we have done the histone modification marker expression. We assayed in the 48 subject, and now we kind of in the process of another 24 subject. So this is for the preliminary, this is only preliminary result. Then addition of the data may change. But for the IL-10 production by monocyte under the LPS, zymosan, or beta-glucan, so those are positively associated with expression of the H3K23AC, which is a typical like a histone activation marker. It's reported to be upregulated in the trained immunity, which is more likely the little more hyperactive innate immune memory. But usually for the H3K27AC is only temporary. Then other type of the intermediate, they tend to have the down-regulated. But at least for the current condition, H3K23AC tend to be continue to be upregulated in ASD patient, which is a little different from the reported histone modification marker pattern reported in the trained immunity, at least mouse model and some of the human studies. So seems a little different from the ASD patient. And we also see that for and it's we did see the IL-10 production is a suppressor cytokine. So we did see we expected H3K23 as AC expression. IL-10 production supposed to be downregulated, so it's going to be a negative association. However, And that was true, at least for preliminary data for the non-ASD controls. But again, ASD subject has a more positive, like, it's not clearly positive. Some of them is, so it's not very, its association seems different. Yeah. But they did see the sum. So at least for preliminary data, those markers for the epigenetic regulation for trained immunity seems like different in ASD patient as compared to non-ASD control. So this is not so many patients because still in the very preliminary, but non-ASD patients appear to have some negative association between the IL-10 production and the H3K23C expression. It's essentially the higher expression, it's lower IL-10 production. But it's more like a positive association we see between the H3K23AC expression by monocyte and then for the monocyte IL-10 production. So that was also a little puzzled us. So we want to see further. Then at least we have looked for the 4 histone modification markers as markers of the histone trained immunity as well as the tolerance type. And the best significant changes were seen in the H3K27AC expression. So therefore, we have now doing the super enhancer chip sequencing, and the target molecule we selected as the H3K23AC. And then this assay is very costly, so that we only checking for the very significant patient of the various influence. So there are 2 different relatively stable ASD subjects, and it's more like a very lot of inflammatory condition and some non-ASD control. And it depends on the result, we try to scale down the little more less expensive, like a little simplified chip sequencing. Then we can do more samples. So, okay. So then this is like a progress report of the AM1. Then the progress report for the second part, AM2, this is we have more data, so a little more detailed result I can show. And this is as I explained, we wanted to have a good, very stable serum marker, and also very easy to be detected by almost everybody. So essentially, we selected from the previous microRNA deep sequencing result, we selected 7 microRNAs. Then we just checked it with a regular quantitative, essentially QRT PCR. Then it's like an expression of the result of the PCR count. It's a normalized PCR count. And I like, and actually now many people now using the normalized PCR count because it's a little small amount for present. These, the way that this is expressed, at least my understanding, is getting popular to express like this way. And also I'm familiar with this, the way that it's expressed because these, the way that a PCR count has been used for the newborn screening for the SCIT. So I'm one of the, I'm involved in the newborn screening SCIT in the New Jersey. So I'm used to be the, this kind of the way expression of the QRT PCR result. And so the essentially we used for 130 ASD subjects and 50 non-ASD controls. And so essentially we didn't find the significant changes from controls of these 7 microRNA PCR count when we combined all the SD subjects. However, when we checked for the changes based on the comorbid condition, there is some changes, and those changes are most significantly observed SD subject with or without seizure or sleep disorders. Essentially, the SD subject without seizure or sleep disorders have a higher circulating level, which means lower PCR count as compared to the seizure SD subject with seizure or sleep disorders or non-SD controls. So this is for the result. Change is not that like a striking, still like a piece. This is Mann-Whitney test result, and there is for the like a seizure negative. There is some MI 423, and also for the sleep positive or sleep negative subset, there is a difference. And it's in general, PCR count is lower in the sleep negative, which means all these the levels of the circulating mRNA is higher. And so, and this is like a 0. Other 3 didn't have shown that the p-value, but it's very close, like a p equals 0.06 or 0.7. So it's a tendency to have this. So this is for the condition, and we did see the some like production and comorbid condition. So is there any problem? So we wanted to see if it was these microRNA PCR count, is there any association with monocyte cytokine production? And especially we thought if this is see some difference, maybe the SD subject with or without seizure and sleep disorder maybe shows different conditions, different association. And actually that was true. We did see some positive associations with production monocyte cytokines. It's a TNF-alpha, IL-6, IL-1 beta, IL-10. So these are kind of the cytokines almost. So that's why I was interested for the serotonin metabolism too. And so this was actually this kind of association is only more evident with without seizure or sleep disorders. The patient who has a seizure or sleep disorders, they didn't have a good association. Essentially, microRNA levels stays low, then cytokine production stays up. That is a type of the patient who has a lot of sleep disorders and seizures. And interestingly, we didn't see such associations when in non-SD controls. So this is for the how it's like associated with. So I didn't see the data for the So I did see the some of the association or ASD total, and this is we just showing the spontaneous TNF-alpha production because this is probably most likely reflecting the in vivo status. Then we didn't show any data with ASD with seizures or with sleep disorder because we didn't see any significant association. And the result was the p-values shown is a result of the Spearman test, but it's essentially similar result we also obtained with Kendall-Taubes test as well. So essentially pretty good association, especially when we see this, when we looked into this association with ASD and without sleep disorders. And this is a TNF-alpha production, and the same kind of the association we see IL-6 production. But it's not as TNF-alpha production, we did see the more good association. And the same way for the IL-1 beta production. But IL-1 beta production, we did see some association non-SD controls. But this is again opposite. This is for the no positive association for the PCR count. It's actually the negative association. So this is again quite a different association that we see with those microRNA, circulatory microRNA levels and the cytokine production. And it's also, again, we did see the spontaneous IL-10 production and miRNA levels. Again, this is for the sleep ASD patient without sleep disorder or without seizure. It's a good positive association. Okay, so the essentially for how if the serum circulatory levels of miRNAs are associated with those cytokine production, then also we saw that some GI, some comorbid conditions, we saw that previously some associations with monocyte production. So we want to see if those kind of if circulating levels of these 7 microRNAs checked, does it change with some of the ASD subject? And this part we haven't completed yet, but at least in 3 patients in whom we are able to get the serum sample repeatedly, and partly because they are relatively closely followed in our clinic and they are sick, so that we are able to get the serum samples. So this is for the patient. Case 1 is really very severe seizure disorders, and we have to use a lot of immunomodulating agent. We used the mTOR inhibitor and also anakinra, IL1 beta blockers, and he was also on the IVIG. And all the levels got better with seizure is actually video EEG completely normal. But after the hit by microbial infection, unfortunately, all the levels went up. And case 2 has been diagnosed with more like encephalopathy, and he has been giving a lot of mitochondrial rescue medicine like leucovorin high dose and IVIG, and also tried on rituximab and a lot of medication tried on the autoimmune encephalitis. But his level stays fairly high levels and never been getting the, PCR values high, means circulatory miRNA levels stays very low. And number 3, these are patients, the third patient is a GI, significant GI symptoms. But he did respond to a lot of probiotics and also have antifungal or other type of the dysbiosis treatment and dietary intervention. And in his case, his clinical features get better, means less hyperactivity, less anxious, and it's more like a more focusing without using any other medication. And it's nicely those circulatory, these 3 circulatory RNAs, microRNAs, PCR count is decreased, means levels are getting higher. So I I think that those circulatory microRNA levels might be the good marker for the disease activity, and especially these levels PCR count high, which means circulatory levels stays low. There will be something problematic or something else we need to look into that. Yeah, and especially I was fascinated with sleep patient who has a lot of sleep disturbance have the very low levels. So those association we need to look into that. So these are not these circulatory microRNA levels are not a good like marker for autism diagnosis, but it may be a marker for neuroinflammation. And also we checked for the what kind of the function these microRNA does, and we haven't completely finished, but it's mostly reported in the cancer study because they are like a suppressor of the cancer. So suppressor cancer means immunologically essentially suppress the immune activation. So those these microRNA is essentially the suppressor of the inflammation. But the number of the articles is much less as compared to cancer research. So we may have to do a little more functional assays. So essentially, this is a summary of the how and the essentially for the result of the circulatory microRNAs, and the low circulatory microRNA, no association with monocyte cytokine. Maybe we can't completely say that, but our data showing that low the microRNA means more like a stays higher monocyte cytokine profile. So I thought there might be something else is and it's retaining for the this high monocyte cytokine profile. Okay. So this is the progress report of the project funded by Brain Foundation. Then also this one is also funded by John Tay Foundation. And the Brain Foundation funding is very helpful because the DHS Department of Health and Human Resources of the New Jersey, they didn't fund us. There is no funding available because of the pandemic. They started funding a little bit this year, so we get a small amount. But we were able to continue this project because of the Brain Foundation funding. And thank you very much for that funding. Okay. And also, I didn't say, but thank you for all the clinical staff and also my laboratory, my technicians and all the collaborators. So that is my my report. So I finish. I think
one question, if you want to answer that, and we can conclude. Thank you so much. It was a great presentation. Thank you, Dr. Blakely.
Yes, the only we have one question. Does dexamethasone or prednisone alter the monocyte marker profile? And is there any benefit to ASD patients?
I think that because of the steroid changes almost everything. So with these assays, we avoided for any of the patient, within 2 weeks of the steroid use, because almost non-specifically all these cytokine production we know it's going to be suppressed by steroid. So.
And I have one quick question. Just that longitudinal profile that you showed, you had, I think, 3 points on it. And I didn't know when that started and how wide those.
Sorry. Yeah, because this is only for the case presentation. The starting point is a time when the patient enters the study. And for the seizure patient, that's before I started treating for the seizures. And then it's like a closely monitored. And so
Okay, and then the inner, what's the distance between your points?
It was it's usually 6 months to 6 months, 3 to 6 months. But each patient has a little different. So I didn't really do that very clearly. But for the because we don't really protocol per se, we're supposed to get to the additional sample for 6 months. So we just followed it. But seizure patient have the striking difference, so that we get it a little early time point. Yeah.
I understand. Thank you.
So thank you so much. And thank you all for attending till the end, and see you all bright and early tomorrow morning at 9 o'clock. Thank you.
Thanks, Pramila.
Thank you.
Yeah, thank you. Yeah.
Thank you.